Thank you for this introduction. Indeed, I'm currently a postdoctoral researcher at Ghent University, where I'm actually focusing on a spin-off project for the time being. And therefore, the title of my lecture is From Polymer Research to Bioink Industry. Now, as you all know, more and more people suffer from conditions or trauma which makes them need donor organs. And unfortunately, the supply does not meet the demand, so this leads to a significant decrease in life quality, or can even lead to a loss of life in general. And what if we could tackle this problem by, instead of replacing damaged tissue, focusing on regenerating it? Now, this thing has always been a topic which I was very intrigued by, so therefore, a number of years ago, being recently graduated as a polymer chemist at Ghent University, I decided to focus on exactly this aspect during my PhD. And then around 4 to 5 years later, actually managed to write this down in a manuscript and got the PhD. So at that stage, of course, there were many interesting topics, many interesting research was done, but the idea was then, as so often is the case, I didn't want this research to die with me graduating as a PhD student, which unfortunately often is the case in academia, that there's great ideas or technologies with great potential never really make it to the real world because basically funding stops or projects are finished. So, and that's a shame. So therefore we were wondering, can we somehow avoid this? So we were thinking, during my PhD, I've been involved in a lot of collaborations with external parties or different research groups using my materials for their specific research. So the idea was, can we somehow make a business out of this so that we can offer these materials in a reliable way, going even beyond researchers, but towards the industry. And then, of course, where do you start? So we started to look at the collaborations. We had collaborations in different fields, being fundamental tissue engineering, regenerative medicine, and drug screening. And there's actually one common denominator there, which is the field of biofabrication or bioprinting. So therefore, we figured if we can offer our materials as ready-to-use bioinks, this will not only bring the research I performed in my PhD towards the real world, but it can also act as a tool for other researchers to have reliable material feed to bring their research to the clinic. So this brings us to the field of biofabrication. Now, what is biofabrication is already discussed by the previous two speakers as well. Biofabrication is basically the use of 3D printing to generate tissues. And what do you need for that? You need to have cells, of course, which act as the building blocks of your tissue. You need a specific design, as also mentioned by the previous speaker, to know where the cell should be and how they should be behaving. And then, of course, you need to make these cells printable. And not only that, they need to be printable, but they also need to be happy and viable. So therefore, you often combine this with other materials, which are referred to as bioinks. So the idea is then that you can 3D print your structure containing your cells, put it in cell culture, and once it's mature enough, you can use it for a whole range of applications, going from regenerative medicine. So this is a bit the dream I think most of us share, that this can be a solution to the problem of a donor shortage. But you could also use it for a lot more. You can use it for drug screening applications or personalized drug screening for specific cancer treatments. And also you can use it to replace animal testing, since you can have human models 3D printed. So that was basically the idea. And then we were looking at which assets do we already have. So we have 4 patents, of which 1 is granted, 3 are pending, which resulted in some materials which quite some unique features. So For example, we were one of the first to have a biodegradable high-resolution resin and managed to process it at the highest resolution ever reported for a biodegradable material, so down to 150 nanometers. We also have a high-resolution hydrogel, which was the first hydrogel enabling cell encapsulation at high-resolution 3D printing. We have first hydrogel which can be processed from the melt, so thereby taking advantage of conventional 3D printing approaches. And we also have a low temperature processable polyester, which can be printed at more cell-friendly temperatures. So these is basically our assets from a material point of view. But then also we have some proof of concept as we managed to use these materials for corneal tissue engineering, development of a blood-brain barrier on a chip, and also to mimic microvasculature. So based on all this knowledge, we wanted to transfer this from the academic setting with the proof of concept towards ready-to-use portfolios for multiple printing technologies, ranging from the conventional deposition-based 3D printing, which is the low-resolution 3D printing, around a few hundreds of micrometers resolution, up to the very high-resolution multiphoton lithography, where you go into hundreds of nanometers. So let us start by the deposition-based 3D printing. So as I mentioned, this has a resolution in the range of a few hundreds of micrometers. And this is the most conventional type of 3D printing, which works. You basically have a cartridge or some kind of a material feed, which feeds your material through a nozzle. And you can deposit it layer by layer by moving your nozzle over the print bed. And then once one layer is finished, you go to the next one and you deposit the next layer. Now, there's a lot of research here, and what is the key feature of bioinks in this segment are the rheological or the flow or viscosity profile of the material. So what do you preferably want is a material which has an easy flow during extrusion, so that it's very liquid to exit the nozzle. But once it left the nozzle, you of course want it to maintain the structure which you've created, so to have shape retention, and of course they also need to be self-compatible. So what you're basically looking for is a material which behaves like icing on the cake. You can easily extrude it and it maintains its shape. Unfortunately, most materials, most liquids behave like this. Once they extrude, they just keep on being liquid, and it's very difficult to print or impossible to print. Now, fortunately, there's some technology which can convert one into the other, which is called shear thinning materials. So shear thinning materials or shear thinning hydrogels are materials which have a high viscosity at rest, so they are very strong or resilient when they're untouched. When you exert a force on that, the viscosity decreases rapidly and you get a very liquid material. So this is what happens in the nozzle. And then once you remove the external pressure, so once they are deposited, the viscosity increases very fast and they maintain their shape. And this basically looks like this. So you can see it extrudes really easily, but it maintains its shape after deposition. Now, a next strategy to have this is to print materials directly from the melt. And therefore we can rely on one of our patents, our Curasol technology, which enables photocross linking of material in the solid state. So therefore we are able to print a hydrogel as a solid and then let it swell afterwards in water, making it the only hydrogel which can be printed in such a fashion. So we built a portfolio around this, which can be divided into two categories. So our first materials is what we are calling our extracellular matrix mimics. These are materials which are suitable for cell seeding, but can also be used for cell encapsulation during 3D printing. And then a second class are scaffoldings, which are only suitable for seeding after printing. And these are typically the materials which are printed from the melt, because of course you cannot blend cells in a molten polymer. So this brought us to a portfolio of around 5 inks for deposition-based 3D printing, where we have the conventional Gelma ink, XGelma inks, which is based on gelatin methacrylamide, which was developed around 20 years ago in our research group, and has quite some good properties as a bioink or just as a material in tissue engineering in general. As this biodegradable, cell interactive, it allows for cell encapsulation. It's a hydrogel which is UV curable, but of course it remains a gelatin, so mechanical properties are a bit conservative, so to say. And we already managed to use this for a whole range of cell types, including vascular cells, brain cells, ocular cells, liver cells, and cartilage, and many others. Now, one of the downsides of Gelma is it can be 3D printed, but it really needs a sweet spot of parameters to make it a bit processable. So therefore, we also offer our next generation of gelatin-based materials, which have the same properties, but are a lot easier to print. And we already have some proof of concepts with adipose tissue and skin tissue. And then our last one of our ECM mimics is a non-biodegradable synthetic hydrogel. It's called XPECT-INX, which is very easy to print. It has a lot of the same features as gelatin, but it's a lot more robust, almost rubber-like, meaning it's suitable for more mechanical, resilient tissue types like bone and skin. And then we have our scaffoldings, which are printed from the melt. So we have our X-Hydro meltings, which is a hydrogel printed from the melt, which can be made cell interactive if desired or not, because for some applications you don't want cell interactivity. It's hydrogel, which is UV cross-linkable and also very robust. And we already managed to use it for different cell types, including adipose tissue, bone tissue, cartilage, and skin. And then finally, we have our polyester-based material X-Solid-INX, which is biodegradable, can be made cell interactive if needed, UV curable, and it's very robust from a mechanical point of view, but also very flexible, thereby resembling a bit cartilage in a way. So we already managed to use this for hair tissue engineering, skin tissue engineering, cartilage, and blood vessel generation. So we also have some distribution agreements with companies, Regema 3D and Felix printers, where you can find our materials. And then, so this is the conventional deposition-based 3D printing. You can go to the high-resolution multiphoton lithography. So multiphoton lithography is a high-resolution laser-based 3D printing technology, which is based on the simultaneous absorption of 2 near-infrared photons, as opposed to single photon absorption in most light-based technologies. And this has the benefit that polymerization can only occur in a very small volume element, which you can scan freely in 3 dimensions, creating huge, well, very high resolution, and also a lot of architectural freedom. Now here the resolution is in the nanometer to micrometer scale, and also here there are some key bioink properties. So first of all, most importantly, they need to be 2-photon active. You want them preferably to be very fast cross-linkable, because of course the higher resolution, the more lines you have to write to generate a construct of the same size. You want it to be compatible, and there can be a benefit if it's solid state cross-linkable, because in this technology, you can freely scan your voxels in 3 dimensions, meaning that if you do this in a solid, your uncross-linked material can act as a support for your newly formed cured material. So fortunately, we can also rely on some of our technology there. So we have a patented Curia Duo technology, which results in faster cross-linking for biomaterials. For example, if we apply this technology on gelatin-based materials, we can process it at 33% faster in comparison to conventional Gelma-based materials. And then the second technology is, again, the solid-state cross-linkability for the reasons I just mentioned. So this brings us to our portfolio there, where we have our hydrogel ink, HydroBio, which is gelatin-based. It's biodegradable, cell interactive, allows for cell encapsulation. It's a hydrogel that can be processed down to 1 micrometer resolution. And we already have applications, again, in different fields. Then we have our Hydrotech, which is our non-biodegradable synthetic hydrogel based formulation, which is cell interactive. It's a hydrogel, it's a lot more robust, and again, can be processed in the micrometer range. And then finally, we have our XPECT-INX, which is our biodegradable polyester-based material, which is very robust and can be produced in the nanometer range. So also here, we have a distribution agreement with our partner at Nano, which are offering our materials through their portfolio. So this brings us to the last one, the LP portfolio. So this is single photon processing where you cure one layer after the other. So key bioink properties there. So this is the intermediate resolution in tens of micrometers. So you need to have material which is photocross-linkable liquid, but also you want to have a low penetration depth, so the light doesn't go too deep that you don't compromise your previous layers when curing the next ones, and you want your material to be cell interactive. Again, here we can take advantage of our Cura Duo technology, and we are currently working very hard in optimizing these formulations, which we hope to present to you soon, still in 2021. So I constantly say we, and we, of course, there's an entire team behind this, which is built around a research group of Professor Sandra van Vlierbergen, which is a professor in polymer chemistry and biomaterials. Then there's me as a polymer chemist, as a project lead and a material specialist. We have Agnes as an application specialist, as she's has a lot of experience in biofabrication and processing materials, mainly via two-photon polarization. We have Aisu as a material specialist, also polymer chemist, Coralie for operational support, Eric as a technician, and Anne, who helps us with the business aspect. So I would like to end my presentation with again mentioning the partners where you can learn more about our materials, and of course also on our website. And if you have any more questions, feel free to consult our website, our Twitter, our LinkedIn page, or send me an email at info@xpext-inx.com, or of course ask your questions now. Thank you.